So the last section in chapter 12 talks about how we actually regulate the cell cycle. So far we've been talking about the cell cycle and all the different steps of it, but now let's talk about how your body knows when is the right time to do these things. So the frequency of cell division varies entirely on the different kinds of cell. Some cells are constantly replicating, like the cells inside your stomach or your hair follicle cells, but some of them won't replicate at all anymore, like your brain cells. And some of them only turn on when they're needed to divide. So it is regulated very, very closely based on the cell type. And these regulations happen at the molecular level. So we're talking about chemical signals that are being sent for the division to happen. It's also based on the genetics. So they need to have these receptors to receive the chemical message to divide or not. And cancer happens when the cell cycle gets out of control, when the regulations stop working and the cells just keep dividing despite your body telling them to stop. That is cancer. We're not going to worry about that. So there's a cell cycle control system which kind of works the same way that a clock does. So basically your cell's going, and it eventually realizes that it's time to replicate its DNA and to make more organelles and then to divide. And so it's like a clock. That's a good way to explain it. And it's regulated both by internal and external controls. So there's things inside the cell telling it to divide, and there's things outside of the cell telling it to divide. Your textbook relates this to a washing machine. It's subject to internal and external controls. So basically, the washing machine isn't going to start until the internal controls tell it that it's filled up with water, but it's also not going to start until you hit the start button from the external side of it. So you need internal and external signals. And one of these are specific checkpoints that the cell, it has it built into it, and it must pass these checkpoints in order to continue. So basically, your cell is set in do not divide mode until you have enough go signals. So that's like every time you pulled up to an intersection, you automatically stopped until someone turned on a green light for you. So just assuming that you're not going to go. So you're at stop until someone says go. So here is the control system, and it's like a clock, like I said. So we start off here. We go through interphase, which is G1, S, and G2, and then we move into M phase, which is a very small part of the cell cycle. And there are three main checkpoints that you need to pass in order to keep going. There's the G1 checkpoint, the G2 checkpoint, and the M checkpoint. And basically, the G1 checkpoint, well, actually, we'll get there. So it seems as if the G1 checkpoint is the most important. For the most part, if you pass the G1 checkpoint, you're going to divide. That's what's been shown based on all the research done on this. And so the cell is automatically going to stop there unless it has enough go-ahead signals, unless it has enough green lights. And if it does, then it generally will go through and complete S, G2, and M phases, and you'll have two daughter cells. However, there's a very particular consequence if the cell does not pass the G1 cycle. What happens is it's going to switch into a different phase that we haven't talked about yet called the G0 phase. And the G0 phase is basically a non-dividing state. So it's doing everything it's been doing during G1, except it's never going to divide unless it gets switched out of G0. So it basically exits the cell cycle. So if at the G1 checkpoint there's a green light, meaning the cell has told the G1 or told itself that it's okay to divide, it has enough signals saying yes, please divide, then it will divide. But if it doesn't have enough of those signals, it's basically at a red light and it's not going to divide. Instead, it's going to move into G0 phase. And G0 phase is basically just when the cell's not dividing. And so these are things like your nerve and muscle cells, they're in G0. They don't divide. And that's why it's really dangerous when you kill your brain cells, because your body they don't divide. They don't make new ones. You kill your brain cells and your brain cells are dead. And that's all. Whereas if you kill some stomach cells, then they will grow back. There are certain cells inside your body, like your liver cells, which are in G0 until your body needs them, and then it'll send those go-ahead signals and it'll switch out of G0 and back into the cell cycle. So the signals that we keep talking about are these things called cyclins and cyclin-dependent kinases. This is a little bit tedious, this information. So there are two types of regulatory proteins. Remember, proteins do basically every job, so these are proteins doing this job again. And we call cyclin-dependent kinases CDKs, because that's a little bit easier to say than cyclin-dependent kinase. And so cyclins are kinases that are dependent, our CDKs are kinases that are dependent on cyclins. So cyclins are these molecules that vary over the course of the cell cycle. And then the cyclin-dependent kinases follow that. They are dependent on the amount of kinases there, or the amount of cyclins there. And one that we're going to talk about mostly is called MPF, which was originally named for mitosis promoting factor. I'm sorry, maturation promoting factor, but now kind of goes by mitosis promoting factor. And it is a, what does that say? Is a, I can't see what it says either. Sorry, guys. Oh. I'm trying. Is a cyclin CDK complex that triggers a cell's passage past the G1, G2 checkpoint into the M phase. Cyclin CDK complex that triggers a cell's passage past the G2 checkpoint into the M phase. So it's only going to go into mitosis if this MPF is present in enough quantities. So here is the fluctuation of MPF activity and cyclin concentrations throughout the cell. So cyclin goes up and goes up and goes up until it reaches its peak during M phase, and then it drops. And then it's slow until S phase, and then it starts growing during G2 phase until it stops. So we don't want mitosis to happen during G1 phase because we haven't even replicated the DNA yet. So it's important that MPF is only growing, or the cyclins are only growing when we want to go into mitosis. And that's when we see the spike of mitosis promoting factor. And basically, this picture's from your textbook. What happens is the CDKs are always there. The orange part of MPF, this CDK part of MPF, is always there. But for most of the cell cycle, it's just CDK and it's not doing its job. It's just a molecule. It's not really doing anything. Then cyclins start to accumulate as we get into late G2 phase. So these cyclins then bind to CDK, and then we have our MPF. So remember, MPF is a cyclin and CDK complex. It only does its job if the cyclin is there. If the cyclin is there, then it's going to promote mitosis to happen. It does that by, it phosphorylates a lot of things. For example, it phosphorylates the nuclear lamina, which breaks down the nuclear envelope, because we can't do mitosis unless the nuclear envelope is broken down. It also helps the chromosomes condense and helps the spindle to form during prophase. But then we get to anaphase during mitosis, and the MPF switches itself off because it starts a signal that degrades its own cyclin. So basically it says, okay, I'm done. I would like this purple part to disintegrate. And it switches on a pathway that allows that to happen. The cyclin goes away, and it's just a normal CDK, and it's not promoting mitosis anymore. So it's a little bit confusing, but basically MPF is only doing its job when the cyclin is there, and the cyclin only appears as we get into late G2 phase. Okay, so an example of an internal control at the checkpoint is if all of the spindle fibers have not attached to the kinetochores of every single of any chromosome. Let's say, for example, I'm chromosome number 10, and I have a spindle fiber attached from this end of the cell, but I do not have one from that end. I'm going to say, wait, anaphase can't happen yet because I'm not going to get pulled apart. I'm not going to get split in half. I'm going to get dragged all the way that way, and then the chromosome numbers are going to be off. So instead, I'm going to set up a signal that says, do not do anaphase yet. Do not initiate anaphase. Do not initiate the chromosomes being pulled apart because something bad is going to happen. So that would be an example of an internal signal. And some external signals are things called growth factors, which are proteins released by other cells that stimulate, or certain cells that stimulate other cells to divide. So the example that your textbook gives is PDGF, which is platelet-derived growth factor, which is a growth factor that is made by platelets, which makes sense. Platelets are the little parts of blood cells that are broken that are part of your blood, and they help clotting happen. And the way that they do that is, or they help your wounds heal. So platelets will release this PDGF, and they will go to cells where you've cut yourself. So for example, say you get a cut on your arm right here. Now, you want new cells to grow there to replace the ones that were killed and the one to attach the two sides of your wound. And so you need a signal for new cells to grow there because normally they wouldn't grow. Otherwise, you would always have a tumor on your arm. So PDGF sends signals from the platelets in the blood to these cells or right around where you cut yourself to initiate cell division. And then the cells will divide and you will heal because new cells will grow in the place where you just wounded your old ones. Now there's a couple of other controls that are external. One of them is density-dependent inhibition, which basically stops crowded cells from growing. So when you get to college, if you're growing cells in a petri dish, you'll see that the cells stop growing once they've covered the whole entire petri dish. They form one layer, and that's basically it. They don't grow on top of each other. They don't keep growing and keep crowding the petri dish because there are external controls that say, wait, we're crowded, stop growing. And basically what that does is it's probably proteins on the outside of the cells. When they touch another cell, they're not going to divide that way because they don't want to, there's only going to be a certain amount of food inside that petri dish for them to grow. So it's a pretty smart move that they stop growing when they filled up the dish. And so that's the same thing inside of your bodies. If there's not space for a lot of cells to grow, they're not going to keep dividing. And there's something else called anchorage dependence, which generally means that your cells have to be attached to something. It says substratum. That would be like any sort of structure inside your body for them to divide. Because if they're just floating around inside of your blood or anything like that and they keep dividing, then you're just going to have a million tumors. So they need to be someplace where they're supposed to be, and then they can divide. And the problem with cancer cells is they do not have density-dependent inhibition or anchorage dependence. So they can grow and grow and grow and grow, even if there's no space for them, which is a tumor, and they can grow even if they're not attached to anything, which is how cancer spreads. So here's normal cells. They need to be placed onto something to divide. So if you just have them in a test tube, they're not going to divide. But if you place them in a petri dish, then they'll divide. Then they grow until they filled up the whole petri dish, and then they stop. And if you take a couple out, they will refill that hole, and then they'll stop. Because once they're hitting each other, they don't want to grow anymore. This is different than cancer cells, which will grow and grow and grow and grow on top of each other and form lots of layers, which is not what we want them to do. And that, we're not going to talk about cancer. That's all.